Thank you very much. Is this on? Can you hear? Okay. Yeah, so I'm probably going to give you a little bit different talk that you've seen for the whole meeting. I'm probably the person, once you come up with these technologies, and you put them in a dish, and, well, first you found them out, then you put them in a dish, then you put them in a mouse, or you create some kind of drug that you want to give to a rat. I'm the person who takes that animal and then does functional assessments on them to make sure that the interventions that you're really putting into these animals work and have an impact. I was going to say on the lives of these rats, but more importantly, and in the end, on the lives of the humans that you're trying to create these technologies for. So if anyone knows anything about sarcopenia, this is the quintessential picture. And this was a term that was coined by Erwin Rosenberg in 1989, and he referred to it as this decline in lean body mass. And he gave us a couple of ways to describe that, but what's really stuck is this idea of sarcopenia, or poverty of flesh. And it happens pretty much to everyone as you age. Athletes, it depends on how much you start with as a reserve, how much you're going to have when you end up, but there's going to be some decline in your skeletal muscle mass regardless. Sorry about that. And here's what it looks like. So, this is my special language that I'm sharing with you today. But what it basically says is this is men, and this is women, this is age, and this is their skeletal muscle cross-sectional area. And all you need to know is it goes down over time. But it's not just the fact that we lose skeletal muscle mass. We also lose strength. And for many years, people have created this little equation which says loss of mass equals loss of strength equals loss of function and disability. But what you can see from this graph here is if we look at just knee extensor strength against lean leg mass in a group of people from the Health ABC study, so we have over 2,500 people in this cohort. Actually, there's over 3,000, but for this analysis, there are 2,500 between the ages of 70 and 79 years old. You can see that there's a nice relationship, but only about 50% of it is described by changes in mass. So there's something else that's going on that is contributing to this loss in strength. So why do we care about this? And everyone puts up that slide that says, there's going to be more people who are aging, and they're going to be in our population. But I like this one because it shows this tidal wave of which way to admissions. So it's going to be a burden on our healthcare system, these aging baby boomers. And this is a topic for another talk and another story altogether. It's also the case that older adults are more likely to have disabilities. So if you look here, this is the percent of people 65, 15 to 64, 0 to 14, with percentage of disabilities, with any disability, and you can see it's at 52%. But the percent with severe disabilities is at 40%, so that's still a huge portion of the population. Now, the process of disability, this is the process of disability that I like to talk about when I'm hypothesizing about behavior and function and sarcopenia and the mechanisms that might underlie this. And what you kind of ignore this for right now, but what I want you to focus on is these measures in humans. And this was put up by a researcher named Naji. And what he said was that there are many things that can start the process of disability going: disease, environment, behavior, or genes. But what it leads to before you see an overt disability is an impairment or a functional limitation in a physiological system. So when you see that, this can become a predictor of who is going to develop a disability. So for example, if you walk very slow, that's very predictive of you progressing to a disability faster. It's also the case that if you have poor performance on these physical performance measures, it also predicts your longevity. So we can look at these measures in humans, but we can also look at them in rats, and this is going to be important a little bit later on in my talk when I use rats as a model for testing interventions and looking at performance. Now, the one hard thing to do with rats is actually ask them about a disability, because more often than not, these are self-reported measures. So you ask a person, do you have trouble ambulating? Do you have trouble getting to the store? Do you have trouble toileting themselves? They can answer yes or no. Rats, can't really do that because they toilet, eat, and do everything where they live, right in their cage. So this isn't always a meaningful measure to look at in rats, but instead we can look at these physical performance measures. And what we see, just as we see in humans, is that there's a relationship between physical performance measures or physical performance in rats and longevity. So rats who have poor physical performance actually die sooner than those that don't. So this is the model that we're working from. And sarcopenia, we believe, is one of these physiological impairments that contribute to declining function and disability, and that's why it's important, and that's why we study it. So this is just to show you, this was a huge meta-study by Stephanie Studinsky showing that, and this is, these are longitudinal data from 65 all the way to 95. Well, cross-sectional longitudinal data in both men and women, showing that survival time over years based on their gait speed. And what you can see is people, so just like we see in rats, people who have a faster gait speed progress to live longer and survive longer. So this is a powerful measure. And it's something that you can actually use in the clinic also. So if you want to look at one of your patients and see if they have a problem, or you want to predict if they're going to become disabled, just having them walk across your clinic is a powerful tool for looking at that and seeing what you can do to intervene so they don't progress so rapidly. Not only does gait speed predict survival, it also predicts other indices of health and function and falls, which again are major contributors and major burdens on our healthcare system in older people. So I'm going to bring up the FDA again because we haven't talked about that enough today. But what is interesting is it's very hard to describe what sarcopenia is. And if you're a company and you want to create a technology, or you want to create a drug or a pharmaceutical that can combat sarcopenia, we can't really give you good information on what it is. And so when a group of researchers who are part of this consortium on trying to determine what sarcopenia is got together with the FDA came back and said, well, we don't want to consider sarcopenia an indication at this time because we don't understand the complex relationship between quality of life and functional outcomes. So even the FDA is telling us that functional outcomes and its relationship to sarcopenia is not known yet. But if you're a physician or if you're a hospital and you want to check a box and give a diagnosis for sarcopenia, and you're a company that has created these technologies and you want to treat, we can't do it yet because the FDA hasn't approved this. So we have to figure out a way to more clearly and closely define what it is. So I'm going to tell you we may be getting to that point where we can do that. Then I'm going to give you another distract you over here and say maybe we really aren't there yet either, because I'm a scientist and I like to do that. But this is the typical model when we create an intervention, that what we really are looking for is what is going on in the physiology, what's going on at a genetic level, and what's going on these cell signaling levels. And then we look at function as kind of a byproduct of what we did. Did it work? Let's just throw a couple of tests in there and see if it hangs, and then we can say our intervention worked. But it's clearly the case also that there's this direct relationship between the intervention and the function. And this relationship is worthy of study, as I've just showed you here from what the FDA said, and because not everybody's going to benefit from intervention, and not everyone's going to have the same kind of functional limitation from this impairment. It's very complicated. I'm going to show you a slide in a minute to show that. So this partnership was put together. This is the Foundation for the National Institutes of Health. And one of the projects that they sponsor, along with the FDA, industry, and several other leading clinical researchers who study sarcopenia, is to try to come up with a way to look at and define what sarcopenia actually is. And one of the most profound things that I think they've said recently is that what degree of low lean mass is clinically relevant. So how much lean mass do you have to lose in order to say that you have sarcopenia? But furthermore, how is it grounded in its relationship to strength and function? So it's shifting the definition from just loss of mass to also how it impacts function. And this just kind of proves the point that over the years, we've been frustrated because we've tried to come up with interventions that build muscle mass. And this is just an example here of testosterone. This is 3 years of administration of testosterone in older men. And this is looking at lean mass, their knee extensor strength, and the knee flexion strength. And what you can see here is that while there's increase in lean mass, there's absolutely no change, and there's actually decrement in their strength. We know this is also the case for growth hormone. You can give growth hormone to older people, you can build muscle mass, but you do not build strength. So we have to change our minds also about how we attack interventions and what kind of interventions we're giving to these people, to older people. So because of this, Clark and Manini, Todd Manini is one of our clinical researchers at the University of Florida, have come up with a different name based on this idea that there's a loss of strength with age, and he calls it dynapenia. And he and Dave Clark talk about this as being, Brian Clark. We have a Dave Clark in our department as well. Talk about this in terms of making the distinction between a loss in mass and a loss in strength. And that these two things could have and most likely do have very different physiological impairments that underlie the result. So the researchers from the Sarcopenia Consortium then, based on this, came up with an idea. Well, here would be our clinical paradigm for a differential diagnosis based on someone who comes in and presents with poor physical function. Do they have weakness? Yes. Do they have low muscle mass? No. Look for some other cause. Do they have low muscle mass? Yes. Then maybe you have sarcopenia. So it's this very simple kind of dichotomous way of looking at what is going on, but it doesn't really get to the heart of why are we still calling this sarcopenia? If they don't have low muscle mass and it's something else over here, then what is it that we're going to call it? And how we're going to diagnose it? And how are we going to make interventions? So this pathway from weakness to cause is still very complicated and not well understood. And I think we have to get off of just looking at this one particular pathway, looking at this muscle loss when we're thinking about interventions, and we have to look at interventions that also engender changes in muscle strength. So this is kind of a paradigm shift for the field, but it's an important one to understand, because if you don't, we're going to keep going down this road of building muscle mass, like giving myostatin inhibitors. So that's a little soapboxy, but that's I really want to emphasize that point, is that we need to look in a new direction besides just building mass to make people more functional as they age and give them a better quality of life. And this is a model people are talking about in the field, is that when we had, you have cardiovascular disease and you have these different other diseases or physiological syndromes that are very predictive on having poor outcomes in these diseases. But what's happened over the years is they've created these cut points. So we know we can monitor these cut points and we can try to control for the progression of this disease. But we don't know this for sarcopenia. We don't know it for dynapenia. And, balance, that may be something else that contributes to this whole change in physical function. So, and we could have many more little lines up here as well that would be major contributors and causes that maybe we don't know all the physiology for. And when I keep saying it's complicated, it is, because Clark and Manini have suggested that this dynapenia is a muscle weakness due to neurological causes. So when you think about that, the physiology of that goes anywhere from the brain to the level of the spinal cord, to the level of the synapses, to the level of the neuromuscular junction. So that's a huge range of physiology that you have to understand in order to get to some kind of intervention if you want to have a targeted intervention for this type of weakness. This is a slide we put together with one of our junior faculty, Thomas Buford, on looking at what all the possible physiological contributions to sarcopenia could possibly be. And I'm not even going to go through this slide. It's just, the title says it all. It's complicated. So I think one of the missing links, so that we can get around some of this complication, is that we can have animal models and natural animal models of aging, which is what we use. So when people mention today, well, rats or mice don't get Alzheimer's, all of our rats and mice age. And they age in similar ways that humans age. And so they become a good model for being an in vivo screen for tests and interventions, as I mentioned before. This was a commentary that was recently in Nature last month. And it was written along with clinical researchers saying that the missing link in looking at studies that can affect longevity and that could also affect health span, so the quality of the remainder of quality of life you have as you age, is using kind of these behavioral functional tests in animals. They particularly were focusing on mice, but we also use a rat model, and we can argue if you want to later if one's a better model or one's not. But anyway, they both have validity and usefulness. And so what you might notice is some of the tests are very similar to what the tests were in humans, and that what I showed you on the previous slide. I was going to show you this swimming test. I have an old and a young rat that are swimming in this pool. Some of you may have seen this a bunch of times before. But it's akin to walking speed because we want to motivate the animal without depriving it in any way. So if we wanted to look at walking speed, and rats, sometimes you have to take away a little of their food to motivate them to run. If we throw them in a pool, they swim really nicely. So we can look at things like swimming speed. We can look at rotorod, so this is a measure of balance and agility, and we can also look at endurance because we can keep them rotating on this rod for a long time and keep them running on it. It declines with age cross-sectionally. We can look at grip strength, as I showed you before in the mice, and in humans this is highly predictive of future disability and mortality. We can look at something called the inclined plane. It's just a plane inclined 60 degrees, and the rat we put it up about 5 feet in the air, and they have to but they have to hold on for, or they'll fall 5 feet onto a bouncy mattress. They learn this very quickly. It only takes 2 times, and we can get a measure of whole body strength. That's the important thing. So what we found is that when we create summary physical performance measures based on some of these tests, and what we find is that animals that are in the highest quartile of performance live much longer than animals in the lower quartile. So again, we have some face validity to our physical performance measures. It's very similar to humans. So what we can do then is we can look at different kind of interesting interventions that are out there, and we can see if we see changes in function or if we can rescue function with these different interventions. And so what we've gone to is looking at ones that are, because this gets a lot of play, is that everyone assumes that if you give a longevity intervention that you're going to have a health span effect. And I would argue that's not always the case. And you better know that before you start giving this stuff to humans, because if you're not going to have good functional outcomes and good quality of life, if they're living longer, that's totally against what we're about. We don't want to make people live longer so they can live unhealthy lives. We want them to live longer so they can live healthy lives. So this is a compound I'm sure you've all heard of called rapamycin, and it's been amazing. One of the most robust interventions, or the most robust intervention for extending longevity in mice across multiple labs when you initiate it at multiple times during the lifespan, males and females. It's kind of unprecedented for this to actually for it to be so strong. So what concerned me with this compound was that it actually blocks mTOR. And mTOR is really important for maintaining muscle viability. And as animals age, they actually have a decrease in mTOR in their skeletal muscles. So if we're mucking with the machinery of the muscle, and then we're compounding it by giving it this compound that actually reduces mTOR, then this could be a problem. So we could have something that actually you have longevity, but you don't have health span. So to me, this was concerning. So and I wrote a paper about it. but it's very interesting because now people are coming back and they're saying, okay, we need to do the work and we need to see if rapamycin actually impacts health span. So you can see a number of studies here, and actually there's a small little, I wouldn't even call it really a trial, there of about 15 older people. I think they're over the age of 90 who are taking rapamycin voluntarily, and then they're being monitored and people are following up with them. They're doing different kinds of physical performance measures in these individuals. So what we found, and I have a lot of groups on here, but if you just concentrate on the green, we found that rapamycin actually helps maintain muscle mass in older animals as they age. So we looked at animals longitudinally during a time when sarcopenia actually hits them the hardest. And what we found is this maintenance of their lean to fat ratio, and we saw a preservation of locomotor activity in these animals. Every species locomotor activity decreases over time, but here we didn't, and you see, but here we did not see that decrease. And I'm sorry, these are young animals and these are old animals, but really what I'm wanting to show you is what's happening in these old animals. But we can talk about the other later if you want. What we didn't see is any change in grip strength whatsoever. None of these data over here in these 25-month-old animals is significant in any way. So while we saw a change in locomotor activity, we didn't see any change in grip strength. And this is in agreement with several other studies that have been published recently, that they're not seeing any change in strength when they see the increases on longevity or other markers of health span in these animals. So what the other interesting thing about this is that there are multiple domains of physical function that you could look at. I mentioned balance before. I mentioned endurance before, and none of these have actually been looked at with these different longevity compounds. So I think that's really important because, as I said before, different people are going to present with different types of impairments and different systems, and they're going to have a different need for a differential diagnosis of what to do and how to help them with their function. So I'll just conclude. I think we need to rethink sarcopenia. Do we even need to call it sarcopenia? Should we have something called sarcopenia and dynapenia? What should we do about this? And we probably need to do something quickly if we're going to, if we want to get treatments into older people for this. And we need to rethink that what contributes to weakness is not always sarcopenia. If it's not sarcopenia, what is it? What are we going to call it? We should embrace function as a predictor and also as an outcome variable to treatments because it's real life and this is what people need and want to maintain their health spans. And that this differential diagnosis, we can use behavior to actually inform the physiology about what is going on in a system. And then from there, we can take a simple assessment of behavior and decide we were talking about tailoring, treatments for people. We could do the same thing by using behavior to tailor a treatment for an individual once we understand this. And animal models are good because they can help us do that. And that's it. Thank you. These are the people, so many people have helped over the years, but these are the people who are immediately working on this with me now. Thank you.